And it's a pleasure today to introduce Bryan Grenfell, who is a professor of ecology and evolutionary biology and public affairs at Princeton University. Bryan began his research career as a PhD student at the University of York in England, where he was trained in population biology, and he actually worked on the population biology of whales. So in terms of scale of organism, he's changed quite radically over the past few years. He then moved on to his postdoctoral work, where he began working on the dynamics of infectious diseases, again from an ecological perspective. He, after his postdoc, went on to several faculty positions, firstly at Imperial, then at Sheffield, is that right? And then Cambridge, Penn State, and then most recently, he moved to Princeton last year. Bryan holds a number of accolades, including he's a fellow of the American Academy for the Advancement of Science. He also holds a medal from the Zoological Society of London, and he's also, just noticed, an Order of the British Empire, which is something that comes straight from the Queen. Anyway, so Bryan is going to be talking to us today about his work on the ecology of infectious diseases, and the title of his talk is The Spatial Temporal Dynamics and Evolution of Acute Infectious Disease. So without more ado, Bryan.
Great. And I first met Paul when he was an undergraduate in Cambridge, and I can tell you all the stories later on. Thanks very much for the invite. It's always great to come here. So what I'm not going to talk about is environmental change in particular, but I am going to talk about non-stationarity. I'm going to talk about the dynamics of particularly highly immunizing infections like measles and to some extent influenza. Measles is a particular case study, which is the kind of C. elegans of epidemiological dynamics, if you like, in terms of its historical data. But I'm also going to talk about measles currently in the Sahel, in Niger in particular, where it's still a major killer. My text, though, really is going to be looking at the effect of external forcing, short-term seasonal forcing, longer-term demographic forcing on the dynamics of these oscillators as a first step. And I'm going to argue that this forcing is quite a handy model if we're thinking about other kinds of non-stationary effects in populations and ecosystems and so on. I'm then also going to talk about more complex infections, particularly like influenza, where immune escape, that is, we saw recently with the influenza pandemic, immunity isn't always that strong. How did these demographic influences interact with immune escape, and what are the mechanisms that really drive that? So I'm gonna go across quite a wide range of things. And there's a large cast of characters, Cambridge, Penn State, our colleagues at NIH, who've been involved in this, and also a number of funding organizations on both sides of the Atlantic. So what I want to do to start with is to talk about the population dynamics of measles, and I apologize to some people in the audience who have seen bits and pieces of this several times, I suspect, though some people won't have done. So measles is a kind of paradigmatic host natural enemy oscillator in ecological terms. We see these lovely biennial epidemics in the UK data. These are just notifications of infection. They notified about 60% of infections, which isn't really bad for an ecological system. Slightly more annual dynamics post-World War II. And these epidemics are driven by by a number of components. And the first of them is really population immunity, and is what one can call the epidemic clockwork. By that I mean we can summarize the dynamics of something like measles, and we don't care, in this case, how many viral particles you've got. We care about whether you're susceptible or infected or recovered. After maternal immunity has waned, susceptible individuals who've never seen the infection become infected. It's a respiratory virus, it's very infectious. And then after a short infection, they moved through to a recovered class, and their immune then to reinfection for life. And the reason for the strength of the immunity is not really well understood, and it's very interesting why it's so strong. Most infections aren't this strong. Then vaccination with an attenuated virus produces an immunity that's also really very, very strong and seemingly pretty long-lived. So if we think about an infection now spreading through a population, then if we think about a pandemic-like situation where you introduce one individual into a big population of susceptible individuals, one infected individual will infect this kind of magical number in my neck of the woods, R0, which is the number of secondary cases that you might cause on average in a totally susceptible population. And this number was kind of bandied about a lot for pandemic flu. Maybe for pandemic flu it was some of the historic pandemics, it was maybe 2 or 3 or more. For this recent one, it's probably 1 point, not very much, not very transmissible. For measles, it was about 20. So you could cause on average maybe 20 secondary cases. There are cases of outbreaks in the Faroe Islands where someone went to a dance and there'd not been infection for 40 years and they infected 120 people in a night. So it's extremely infectious. What happens then, though, is that in a kind of predator-prey sense, this rapid increase in the number of cases depletes the susceptibles really quickly, and the births can't really keep up with that because that's a kind of linear thing, and therefore you get a rapid depletion, and this is this indirect effect of herd immunity. So the epidemic turns over at a kind of critical proportion of susceptibles, and there's a lot of mathematics that can be expressed, but essentially that picture captures that. So this is the first component of the dynamics, which is really the epidemic clockwork, but then there's a bunch of other effects which influence the dynamics of measles, and these are the dynamics of measles again in England and Wales here, similar to what we saw before. Onset of vaccination, and we see much more irregular dynamics and a gradual trend downwards as the authorities gradually got their act together and increased rates of vaccination. But before telling you what the other components which drive the dynamics of this ecological oscillator are, I wanted to just show you some observed data, because the other nice thing about these systems is that there are really great data. So these are historical data for England and Wales. The areas of the circles are proportional to cases. London is in yellow, and then smaller places are in shades of orange. And yet London's yellow here, and it's scaled onto 0, 1 here so that we can see it compared to the rest of the country. There's a kind of annual period at the start, and then there are these lovely, at least to the eyes of the dynamicist, biennial oscillations. So very, very, very synchronized biennial oscillations. And you see that there are big epidemics. There's a big epidemic every other year. In terms of our threshold, our clockwork, it took about 2 years, almost exactly 2 years, for the birth rate to top the susceptibles up to a threshold where there could be another epidemic. But then you see in between the big epidemics, there are small epidemics as well. And I'm going to come back to that seasonality. So now, and the other thing that we see here is the spatiotemporal dynamics. And we and a lot of other people have done a bunch of work on this. I'm not going to focus on spatiotemporal dynamics today. But there are strong waves of infection moving away from the big places to the small places. And the infection kind of winks out in the small places due to demographic stochasticity. Small numbers of cases in troughs means that, you might, the crucial 3 people might not go to the supermarket, and therefore the chains of transmission might break, and therefore has to be started from big places, so that's kind of forest fire dynamics that drives those oscillations, oscillations in space. Then when we move through to the vaccine era, things get much more desynchronized, and that's again another story, and much more irregular in their dynamics. But you see that another reason why people have looked at this stuff a lot, apart from its public health importance, which I'll come back to later, is that the data are really great, and the natural history are relatively simple. So anyway, there's a bunch of other effects. And I'm not going to, and there's again a lot of work on this, but it's published, and I can point you at the papers if you're interested, that influence the dynamics. So the recurrent epidemics, by that I mean the epidemic clockwork that we talked about, this topping up of susceptibles and then using them up very quickly by the epidemic. There's demographic stochasticity, that is, the epidemic disappears in small places in the troughs between epidemics, doesn't disappear in big places. That threshold was about 250,000 in these UK data. So in cities above that size, the infection always kind of tottered along in the troughs between epidemics. The other effect is seasonality, and we'll come back to this, but that's effectively the seasonal aggregation of kids in schools, which acted like two people pushing a swing for the mathematicians in the audience. That this is like a damped pendulum in its dynamics, and if you push it at either end across the biennial cycle, then you'll force the oscillations to be sustained. And then there are long-term secular changes, which are vaccination, obviously, but there are also changes in the cyclicity, which I'll come back to and explain in a minute. So there's a bunch of, if you think of a kind of a dynamic analysis of variance, there's a bunch of drivers that push the dynamics to different extents. There's also a complicated story about those waves and spatiotemporal dynamics, but that's not germane here. And just to say, and I'm not going to go into the details of this because I want to get to the more applied questions, we can use time series analysis to look at the non-stationary dynamics here. Here are the dynamics of measles in London, just on a log scale to make them a bit more sinusoidal. And we can use wavelet spectra, so we look at power as a function of time as well as frequency, and we can see the strong biennial dynamics there, and then a gradual change in period in the vaccination era. So there's a massive kind of time series analysis that we can do on these sorts of data. So that's kind of frequency domain time series analysis looking for patterns. So then you have to do process, and to do that you have to fit those SIR models to the data effectively to estimate R0 and the seasonality. We can do that with state-space models, so we can fit to step-ahead predictions of the model, and the step-ahead predictions, this is of the number of cases next generation, which is about 2 weeks, is a function of the susceptibles now and the cases now, depending on the seasonality of transmission. And we can fit models to capture that seasonality of transmission. And to cut a long story short, they're quite successful at capturing, this is for London, the biennial dynamics through much of the pre-vaccination era, and the annual dynamics earlier on. And the annual dynamics, the other ingredient of this model is birth rate, so that's the fuel that's driving this oscillator, that's pushing it back and forth. In the baby boom post-World War II, at high birth rates, you tend to get annual dynamics. So that's the non-stationary. So this is kind of like reverse vaccination, if you like. It's increasing the birth rate, which pushes the dynamics and means that the threshold is hit more frequently. So that's how we get at the kind of mechanisms here by fitting these models. So what about the dynamic impact of vaccination, finally? Vaccination acts like a reduction in birth rate, effectively. And this is just to illustrate, and I think it's a great illustration for public health people, actually. If you, and this is not our work particularly, and it's a bunch of work by previous people, particularly Dieter Schenzler and Joan Aaron, and then we did some work with David Earn on it. If we just take our seasonally forced measles model and we vaccinate, and these are two different runs, and this red case is vaccinating in the epidemic trough. We get sort of slightly longer period, slightly complicated dynamics. If we vaccinate in a peak year, as the epidemic's going down, we'll get completely different dynamics. So there is the possibility of exotic dynamics here, coexisting attractors, for example. Depending when you vaccinate, when you perturb the system, you might get different behaviors. This happens, incidentally, because if you vaccinate in a high year, it's about to be followed by a deep trough. So you're really exacerbating that trough effect and pushing it into a different regime. And if we fit the dynamics with our time series versions of those SIR models, we see that these relatively irregular dynamics, so you fit to the pre-vaccination era and you kind of project forward, it's still messy, and you have to allow for kind of extra vaccination that happened in 1968. They did some kind of catch-up vaccination when they started vaccinating of older kids. If you allow for that, you get a reasonable picture of qualitatively what happened, but it's very hard to predict the irregularities because of those coexisting attractors, because of the effects of stochasticity. So prediction, even in this seemingly very predictable system is actually pretty tough. So really what we're trying to do is project what the dynamics are going to do. So one final ingredient. So far I've talked about the forcing of the dynamics relatively gently, relatively low seasonal amplitude. There was tremendous excitement in some parts of the ecological community about 20 years ago, particularly Bill Schaefer, which is a tremendously stimulating incursion into this area, looking at some of the irregularities in the dynamics of things like measles and interpreting them in this way. So this is a bifurcation diagram, and what we essentially do in each column of this figure is we pick a seasonality and we just run a deterministic version of something like the model that I showed you, and we look at the number of troughs. That is, we look at the period of the cycles and the richness of the dynamics. So at very low seasonality, you get annual dynamics. There's only one sort of trough. At moderate levels of seasonality, there are biennial dynamics, so there are deep troughs and shallow troughs and deep troughs and shallow troughs and so on. But crank the seasonality up a little bit more. This is a forced oscillator, and you get chaotic dynamics and very, very irregular. And a lot of people got very excited about this, but a bunch of us, and I was one of these miserable people, pointed out that this is 50 million people in this simulation, and this is rather a deep trough. Okay, you can't simple chaotic dynamics in this system really won't explain what we see, and it's much more variations like these limit cycle behaviors and changes in cyclicity because of birth rate and so on. So it seemed that these exotic dynamics were irrelevant. Now, is that really true? So to explore that, I'm going to talk about a collaboration, and this is a lot of this work was by Matt Ferrari, who's now on the faculty at Penn State, who talked about a piece of this a couple of years ago at the School of Public Health. And this is with our great collaborators at Epicentre, which is the kind of research arm of Médecins Sans Frontières, who have great links with Niger. So we wanted to look at dynamics and vaccination of measles in the Sahel. Measles still kills some hundreds of thousands of children a year, a lot of them in this Sahelian region. So we'd like to now apply our models from developed countries and try and look at optimal vaccination strategies here, because it's not possible to get the very high levels of vaccination that would allow one just to interrupt transmission for kind of health system reasons. First thing we need to do, though, is we need to understand in general what we might expect from the previous work about epidemiological dynamics at high birth rates. So Niger has very diverse, most of the populations along the southern edge of the country. Northern Nigeria is down here. Very, very high reported birth rate. 50 per thousand, like triple the birth rate that we saw in those historical measles data for the UK. A relatively low vaccination coverage. So what's our expectation just from taking the simple models that I talked about, cranking up the birth rate? And this echoes a bunch of work that was done about 20 years ago by Angela McLean and Roy Anderson. And what they pointed out was that if you crank down the birth rate, you get rather irregular dynamics. That is, if you vaccinate, for example, as I've discussed. But what I want to focus on here is if you crank up the birth rate for the models that we developed and fitted reasonably well for the UK, for example, or the US, you get very, very regular annual dynamics. And you see the troughs here are very, very shallow, so that you would get strong persistence. The population would now persist. Measles would now persist in a population of 100,000, not 250,000, as I said before. So that's the prediction, very regular dynamics. If, in fact, we look at the seasonal dynamics of measles in Niger, where there are absolutely magnificent data, then there are indeed annual patterns nationally, but of course one needs to disaggregate. And if we disaggregate by district, and let's focus on Niamey, the capital, then in fact we get nothing like what we expect from the model, so the model's wrong. We get irregular dynamics, very, very spiky epidemics, lots and lots of fade-outs, lots and lots of missing epidemics. So very, very different from what we see. Very erratic epidemics, very variable around the country. And the reason is, and this is a longer time series for Niamey, and Niamey has a population of 700,000, so it should persist easily. Measles should persist easily. It disappears after all these major epidemics, in fact. And it's because if we fit those models that I talked about, those SIR models, to the data, to the time series, we find that this thick line, this is transmission rate. This thick line is the seasonal swing of transmission rate for those old UK data. And this is the seasonal swing for near-May. And it's much, much more seasonal. And that seasonality is focused, that most of the transmission is focused in the dry season. And the supposition here is that, as far as we can tell so far, and I'll come back to this, this isn't to do with schooling at all, because the median infection age here is about 2 or 1. It's to do with families moving in and out of the cities associated with our annual agricultural migrations. We've not got the seasonal disaggregation of birth rate yet, but so it could also be birth rate variations associated with that. The seasonality in birth rate could also be a driver. So it forces very strong local extinction. So let's use the models to look at that a bit more systematically. And this is another bifurcation diagram, and this is how you read it. As before, I'm looking at the dynamics as a function of the strength of seasonality, but I'm adding an axis of birth rate now, and I'm color coding. So we run our simulations, and we look at the cyclicity that we see in the simulations as a function of birth rate and strength of seasonality. So these are the data for the seasonality, the low seasonality amplitude for London that we saw, and you see that all we see there are annual and biennial epidemics. It's very strongly locked to annual and biennial patterns. And that baby boom effect would, for example, be as you crank up the birth rate, you might move from biennial to annual epidemics. If you crank the seasonality up to the level of near-May, you in fact get much more, in general, irregular dynamics, and over much of the range, you get chaotic dynamics. You get extremely irregular dynamics. And that's because there's this incredibly strong seasonality. Now, one shouldn't get excited here about chaos rearing its head here, really, because these troughs are very deep. This isn't simple deterministic chaos. The measles would go extinct at near-May's population size in this hatched region. So what we're really talking about is kind of somewhat deterministic instabilities in the dynamics, leading to these very, very irregular patterns. So what are the implications for vaccination and for the uncertainty of the system? So I'll show you this again. So by uncertainty, I mean, if we now think about cranking up vaccination rates, which, remember, is a bit like dropping birth rates, okay? Cranking up vaccination rates in near-May, then you're moving down in this direction, and then at some point the infection will wink out because you'll hit the herd immunity threshold. Then it's going to stay irregular right down to the edge of extinction of infection. So what's going to happen is the dynamics are currently like this. So you might have irregular epidemics every few years with fade-outs. What you would have got in the current UK case, where vaccination rates are not really fantastic in some groups, you get a low grumbling infection because it's weakly seasonal. What you would get in these regions is nothing very much than an extremely violent epidemic, and the more violent it is, the higher case fatality rates tend to be, for example, then a long gap, and then an extremely violent epidemic. So we're currently interested in, at least as a theoretical exercise, the way that you give the extra vaccination that's needed in these regions is by supplementary immunization. So you go in and you, as well as routine vaccination, you go in with a supplementary campaign every few years. In theory, if one could pulse those campaigns of vaccination, maybe we can entrain these dynamics. I think that's a theoretical exercise, because really what one needs to do is ramp up the background vaccination. And the key point here, in an applied sense, actually, is, this is a great vaccine and it's cheap. And we really should be keeping the vaccination ramped up in these regions. And that's really the issue. But things will be deterministically unpredictable. So, in fact, Niger, population of kind of about 20 million, is is probably not at all the regional driver here, because measles probably, if Niger was kind of isolated, measles would probably go extinct for long periods completely across the country. The driver is northern Nigeria, where we're just beginning to collect data, and it's this oscillator that you really need to understand, and it's probably regional coordination of vaccination that's going to help us quite a lot there. But for measles and for a lot of systems, and we're particularly interested in this, and I'm in particular, my postdoc Nita Bharti is interested in this, it's the population movements, therefore transboundary population movements, that we're really interested in. And I think cell phone data, as cell phone usage spreads in this region, and it's still not that high, is going to be very useful there. But another thing that Nita and Andy Tatum at University of Florida is looking at is night lights. Now, night lights are surprisingly effective these days if you correct for mooned or moonless nights, clouds and so on, at spotting fires and so on, so that we can capture crudely roughly what the population movements into and out of the cities are seasonally. And what we tend to see is that there are density changes that are consistent with our population movement things. So in the dry season, trying to correct for everything else from the images so far, then you see stronger night lights in these red regions than in the wet season, and that seems to indicate an increase in density. But what we really need to do now is get more data on the seasonality of birth rates, look ground truth more explicitly how people move, so that we can get at why this seasonal oscillator is so strong, and then also transboundary movements. And if you talk to the people who work on malaria or many, many other infections, movement patterns are really key. And I think that, it's a very interesting kind of cross-disciplinary problem. And then finally, for many regions won't have good data as good as there are for Niger, so it's interesting to wonder if one can then take patterns of movement and patterns of seasonal aggregation of people across Africa, for example, across this belt, and think about how measles co-varies with the agricultural season, and try and make larger metapopulation models. And that's an aspiration for the future. But you've really got to get at the movements first. Big interest currently, and again this is mostly Matt Ferrari's work, is that the local dynamics of access to care in an applied sense is absolutely key. By that I mean it's all very well improving the supply chain or even getting better vaccines and getting the vaccines to clinics, for example, but you've also got to think about how easy it is for people to access those clinics. So a bunch of work that we've been interested in recently is how in rural areas vaccination rates are much lower, and how, because people just can't get at the vaccine, and thinking in future about how one might optimize that access in these regions is going to be very important, because again it's a way of fighting this seasonality. So just to summarize the measles part then, we've seen the epidemic oscillator. We kind of played in that sandpit of nonlinear dynamics in ecology. There are seasonal and demographic forcing these non-stationary effects, particularly demographic changes and vaccination changes. I wonder if the main impact of environmental change here, if we want to think of that in terms of cholera, or we want to think about it in terms of malaria, then there might be direct drivers environmentally, and there may be climatic drivers of measles dynamics seasonally, but it doesn't seem that that's the case currently. But I wonder if the major effect on these directly transmitted infections is going to be by things like trends of urbanization, trends of population movement, as environmental drivers change. And I think trends of nutrition, perhaps, and I think that's going to be an interesting area for future work and growth in this area. So what I want to do now is confess that measles is in fact the exception. A few of these childhood infections have extraordinarily strong immunity. Most infections are, in quotes, much smarter than that. Immunity is much more imperfect. TB is a classic example. HIV is, in a different sense, much more chronic infections. But even acute infections like influenza, there's considerable patterns of immune escape. I just want to illustrate roughly what I mean by that So if we compare measles and flu, they're both RNA viruses. They both have quite a large error rate in the polymerase. So there's a lot of variability generated. So there's a lot of basis for immune evasion in terms of variation. But if we think about the susceptible to infected to recovered class for measles, then each of these is supposed to be a strain of the virus. But so far, it seems that cross-immunity is incredibly strong. Any strain, vaccinating or having natural infection of any strain of measles will protect you against any other. There are even quite strong cross-species effects for the other vaccines for other morbilliviruses in the measles group, like canine distemper, for example. Very strong cross-immunity. That means that all these within-host intricacies that would produce variations in the virus don't matter, because you can chuck them away, because it's the same recovered class. Okay? So that means you get a simple oscillator, you get these multi-annual predator-prey type dynamics, and you get rather a boring phylogeny for the surface molecules with just some spatial variations. And thanks to Eddie Holmes for these viral phylogenies. For flu, as we know, then just to cartoonize it, there's a progressive change every year, or in many years at least. So therefore we get a kind of ladder-like phylogeny, rather uncertain population patterns, complicated population patterns. Herd immunity is a rather slippery thing for flu because the virus is changing all the time. But all the patterning is really in the phylogeny. So very, very different patterns. And now I'm going to think about how and there's a bunch of theoretical work trying to explain how patterns of influenza change through time and how the dynamics and the evolution are related. And maybe the H1N1 pandemic will tell us more about that. But what I want to do is step back from that and think about how our analysis of demographic drivers and how they push measles dynamics are affected if we think about the dynamics of these imperfectly immunizing infections. Now, we can think of influenza as really an SIRS infection. So very crudely, you're now susceptible, you're infected, you're recovered from infection, you're immune, but then you go back into the susceptible class, very crudely. Okay? So how's that going to affect our coupling of birth rate to dynamics that we saw for measles? Well, we know that the dynamics of these very strongly immunizing infections, like measles, promptly track the dynamics of the infection. So if you change the birth rate, you will change the dynamics. And we saw that very clearly in practice and in theory. If we think about this now as a kind of environmental oscillator, and we think about this big flow, so there's a small birth rate there relatively, there's a big flow if immunity only lasted a year or two, for example. There's a huge flow of individuals back into the susceptible class. So that's like a big averaged birth rate. Okay? So if we think of this as a kind of oscillator of some sort driving the system, then this is a much more powerful oscillator kind of swamping it. So if I simulate that, here's a simulated baby boom. Here's the effect on measles dynamics, and we see that there's a nice tracking of the dynamics in terms of the epidemics. If I now assume, and this is just an analytical interpretation of this, which I won't go into here, if I just assume that same model, but the immunity only lasts a few years, then the same baby boom has no effect. So this is a kind of demographic buffering, effectively. If infections don't track birth rate, then it's an implication that there's very strong imperfectness in the immunity. And I'm not going to go into details here, but if we look at the dynamics of the simple models here, then this is really very much like if we look at the susceptibles, that is the prey against the cases, that is the predators. This is very much like the Lotka-Volterra model for predator-prey dynamics. This is for the population dynamics folks in the audience. And vaccination and the tracking of birth rate in a vaccination is very much like the paradox of enrichment. So if you vaccinate or you reduce birth rate, then you'll reduce the number of cases at equilibrium, but you won't reduce the proportion of susceptibles. And that tracking of birth rate of the dynamics as you change the recruitment rate of susceptibles is very much like the paradox of enrichment in predator-prey dynamics. Talk about that in more detail if people are interested later on. But if we look at SIRS infections, then at different birth rates or vaccination rates, there's no change. So the equilibrium, which is the intersection of these nullines, really sits there because effectively, if you vaccinate or you reduce the birth rate, you'd still get a lot of people coming back into the susceptible class. So there's much, much more buffering of the dynamics. So if we're thinking about now demographic change or environmental change, the same thing applies. And we're thinking about perfectly immunizing infections should track that really strongly. Imperfectly immunizing infections shouldn't track it. And here's a possible instance, which is respiratory syncytial virus in the US and against plotted on the same axis here as another imperfectly immunizing infection, para-influenza virus. And there's a trend associated with birth rate in this case, but not in this case. So this may be an instance of that. But we've not got a conclusive example. And one possible example, a very immunizing infection, is rotavirus here, which is a major human diarrheal virus. And Ginny Pitzer, who's used to be in Harvard and is now in my group and gave a talk a couple of weeks ago about this. Indeed, this does track the birth rate very strongly, rotavirus. So, but for example, influenza does so, I think, much more, rather more weakly. So imperfectly immunizing infections should really track birth rate changes and environmental influences much more gently than imperfectly immunizing infections, the theory tells us. So a corollary of this demographic tracking is that if we look at the theory, transmission must be at least partly density dependent. If you increase the density of susceptibles, which effectively what you're doing by increasing birth rates, that translates through via this kind of paradox of enrichment thing into it translates through into more predators, that is into more cases very, very promptly for very strongly immunizing infection. So there must be a density effect. So therefore, the R0, the transmission rate, must kind of track host density to some extent. Now, that's certainly true in a complicated way, which I can come back to if you want to discuss it, with childhood infections. Certainly true of smallpox. Towards the end of the smallpox eradication, high-density countries, they had to reach higher levels of vaccination uptake. But it's not always the case. And this is just to give an advert for a piece of work with our collaborators at the Marine Mammal Center. So this is an animal disease example. And this is our wonderful collaborator, Francis Gulland, who's collected data from these glamorous beasts, Californian sea lions, of leptospirosis, which is a major human zoonotic infection since the 1980s. And these are haul-outs of infected animals. And you'll see that there are these fluctuations, and it seems that it's an endemic infection. But all I'm going to say about this is this is a different example, because in this case there are massive changes in the population. This is pup recruitment, which has shot up. The population had recovered since the 1970s. It had been threatened then, and population size has changed similarly. And El Nino, particularly this El Nino, had a very, very strong effect on pup recruitment. But if we look at R0 as a function of population size, then there's no trend. This is frequency-dependent transmission. It looks as though densities of populations, local densities, stay pretty constant across a wide range of recruitments and population dynamics. And therefore, in this case, the dynamics kind of carry on regardless of the disease as a function of population size. Now, that doesn't matter in any applied sense here, because this is a very healthy population. But I don't know if you know about a wonderful example of a very exotic infectious disease recently, which is a transmissible cancer in Tasmanian devils. Wonderful work by Hamish McCallum. That's a frequency-dependent transmission, it turns out, probably because it's somewhat sexually transmitted, aggressive contacts and associated with sexual transmission. And in that case, it's the kind of reverse of this. The population, the transmission rate and the death of these animals from the cancer will stay high even as densities decline. So it's a doomed population unless they take major effects. So this effect of frequency versus density dependent transmission is a very important modulator of all that I've talked about. And I should say that's a paper just about to be submitted by Jamie Lloyd-Smith and Kim Pepin and a bunch of other people. And one of these folks would be great to come and talk about this. It's a lovely story. So finally, I just want to drill down into some detail just to explore an interest that a bunch of people have got in this audience, and I'm interested in it as well. Ultimately, if we're going to understand these infectious diseases, despite what I say, for infections like flu, certainly, we've got to drill down and look at mechanism more. We've got to get down to some extent, the within-host dynamics and look much more mechanistically at the evolution of immune escape. Now, that's a complicated business. Here's the flu virus with the hemagglutinin and the neuraminidase, which are the antigenic surface proteins. And in particular, the hemagglutinin gets you into cells, roughly speaking. Neuraminidase gets you out of cells if you're a virus. And both of them vary, and the hemagglutinin is the one that varies over time, as I described, and that's the thing that drives the pandemic changes and the seasonal influenza changes in particular. But if we think about if we really wanted to understand this, what data would we need? Because data are the key thing here. We'd need data across scales of the dynamics of mutation, of viral shedding and immunity in individual animals, transmission, the epidemic dynamics, and in particular, founder effects in epidemic troughs, regional dynamics, and kind of global phylogenies. Now, this is a keen-eyed audience, and you'll have spotted that these are not humans. You can't do this in human systems. You have to have animal systems. And the horse is by no means an ideal experimental animal, but it's a useful one in this case because it does have a global phylogeny, because it harbors an endemic infection of H3N8 influenza. In fact, recently jumped to dogs in the US and the UK, interestingly. And because thoroughbred horses are much more looked after than humans, they're extremely valuable, there's a lot of work testing vaccines and looking at this level. So I want to look at the calibration between immune escape and determinants of transmission, just to give an illustration of the sorts of data that we need at these different scales. And this is me standing next to this pony. It wouldn't let me get any closer than that, actually. So these are Welsh mountain ponies at the Animal Health Trust in Newmarket in the UK. And these are my collaborators. This is a model horse. And these animals, the nice thing is they could definitely guarantee from sort of isolated farms, and they would serologically test them before the experiment, you could get naive animals. So you could actually have controls to some extent when you did infection experiments. And the infection experiments were actually testing vaccines. So the way that they tested the vaccines was to immunize naive animals with a vaccine, and then challenge the horse with the same vaccine or a different vaccine at a given distance in terms of number of amino acids in the viral hemagglutinin. So you let the antibodies drop off a bit, you challenge with the same virus or with a slightly different or a very different virus, and then you see how determinants of transmission vary. Or at least we did that when we reanalyzed these data. And no animals were hurt in this course of this experiment. These are two of the animals. Usually experimental animals are killed, but these were given to small children, I think. It was a kind of unusual exception of the usual rules. And what we see, if we look at amino acid changes between the challenge virus and the immunization, so this is the same virus, more or less, and this is the control, this would be like a pandemic virus. This would be at a kind of infinite distance. So this is if you just have a naive animal at the start. Then these are various correlates of R0. So the probability that you get infected, that you seroconvert, the probability that if you seroconvert, you excrete the virus, and the period for which you excrete it. And you see they all increase so that the so that these components of transmission rate are increasing. However, it's important to stress here that they didn't directly measure transmission. You didn't take the horse and put it with 6 other horses and see how many it infected, okay? That would have been the kind of holy grail experiment here. And I think people are doing this now with guinea pig models and so on, and ferret models, to get at this a little bit more explicitly. So if you put that into a population model, you see that it takes about 4 amino acids in the active sites of the hemagglutinin to allow the effective reproduction ratio to go above 1, and that means each case causes more than 1 case, and then you would get a big epidemic. So these are qualitative results, and it's an animal model and not the human infection, so you wouldn't make specific extrapolations here, but it's a point that to move to the next generation of epidemic models and to think about, you know, the long-term effects and control of epidemics, we're going to need to drill down into these kind of immunological depths and to think about immunology, microbiology, virology. So overall conclusion, I've looked at demographic drivers, human movements, thought about measles. There's a whole other spatiotemporal story that I've not talked about. And the big challenge is combining evolutionary and epidemiological dynamics. So rotavirus, which Ginny talked about a few weeks ago to some of you, and flu probably show similar individual level dynamics of immune escape, kind of. There's immune escape, there's heterologous and homologous immunity, but incredibly different epidemic patterns. And understanding the kind of zoo of patterns and of viruses at the population level and how those relate to the kind of within the kind of cross-scale dynamics is a kind of really interesting and important area, I think. Okay, thanks.
Thanks, Brian. So we have some time for questions. Scott.
In the flu
Oh,
example.
sorry, Scott. Would you mind actually stepping up to the mic?
You want to record me?
They want to record you. Yeah, for posterity.
Hi. I was just wondering in the flu example at the end, whether you saw any evidence for interactions among mutations. In other words, you sort of had a count of genetic distance between the vaccine, but is there any synergistic effects of mutations?
It's a great question. We looked and we couldn't, and this is mainly, I should say, my work by my colleague Andrew Park, who's at University of Georgia now. We looked and the design wasn't kind of balanced, you know, so we didn't really see those effects. This was just published in Science, and the next paper in the issue was a wonderful paper by Hinsley et al. By folks from NIH, where they looked at immune escape in a mouse model for flu in chains of naive and partially immune animals, and looked at how that co-varied with avidity of the hemagglutinin for the cell. And, you know, that's exactly what you're talking about. But there's probably all kinds of compensatory mutations and all kinds of stuff going on. You're right. It's a good question.
Any other questions for Brian? I have a question sort of following on a little bit from Scott's, which is, to what extent do you think there is ability, if we, you know, you talked about probing into the depths of mechanism, that in some sense by doing that, you will ultimately be able to say something about either the predictability or the unpredictability of there being an outbreak sometime in the future. For example, you know, in terms of the evolutionary process, how much is it gradual evolution versus punctuated equilibrium type dynamics?
Well, I was about to give a pessimistic answer until you came to the last sentence there, actually, because I think predictability is really hard here. But I mean, and if you could, in seasonal flu case, and a lot of people are working on this, if you did know enough to predict where it was going to evolve next year or in the next few years and could tailor the vaccines, this is not a transmissible virus, so that would be a very powerful tool, actually. But in terms of punctuated versus gradual, flu is kind of the classic example where we do know that, right? So gradual is seasonal flu, and we don't quite understand how it works, but we have a rough idea. Punctuated is recombination, is reassortment, and is something jumping out of birds or pigs and so on, you know. So we have a qualitative idea of that. Understanding when, you know, predicting when pandemics are going to happen, you know, good luck, really.
Any other questions? All right. Well, we'll thank Bryan one more time.